Be welcome to the session of the Chemical Engineering Seminar. We have as a guest Dr. Julianne Holloway. Julianne Holloway is an assistant professor of chemical engineering at Arizona State University, ASU, and an associate faculty member within the Biodesign Institute's Center for Molecular Design and Biomimetics. Prior to Arizona State University, Julianne completed her PhD in chemical engineering at Drexel University and her postdoctoral training at the University of Pennsylvania. Julianne's research group integrates biomaterial design with innovative manufacturing to control and direct stem cell behavior to tissue engineering and regenerative medicine applications. Julianne is also committed to service, including recent election to the American Institute of Chemical Engineering's Board of Directors, serving on the editorial board of Regenerative Biomaterials and as a past associate scientific advisor for the Science Translational Medicine. Her contributions have been recognized through several awards, including ALKE's 35 Under 35 Awards, ALKE's John C. Chen Leadership Award, Mayo Clinic ASU Alliance Faculty Summer Fellow, National Institutes of Health NRSI Postdoctoral Fellowship, and others. The name of the seminar is Designing Biomaterials with Spatiotemporal Control for Tissue Engineering Applications. Please be welcome, Dr. Julianne.
Thank you very much for the introduction. And as mentioned, today I'm going to talk about the work that my lab's been doing to design materials with both spatial and temporal control over their properties for various tissue engineering applications. To give you a quick overview and background in terms of biomaterials and how they've evolved over the past few decades, you can think about what are called first and second generation biomaterials as the traditional metals or polymer kind of joint replacements. These are non-degradable materials, so they're meant to replace and to mimic biological function. Over time, we've really started to design biomaterials that don't just replace tissue, but also directly interact with the surrounding tissue, and perhaps even try to encourage those cells to regenerate tissue. And so that instead of having some sort of synthetic replacement, you can actually encourage full tissue repair of the diseased or injured tissues. And so you can see that these biomaterials have increased both in their complexity, perhaps they are composed of multiple different materials, as well as their bioactivity. And so when we think about how you design materials or approaches for tissue engineering, typically there are three components that you can think of. The first one that I'm going to talk about is cell source. So these can be stem cells that can differentiate into different types of cells, or already differentiated mature cells. You could design systems where the goal is to deliver the cells into the wound area, and those cells participate in the wound healing process. Or you could also try to encourage cells that are already in the body to migrate to where the wound is. You can also think of the signals. So these are the molecules. They can be small molecules or large molecules like proteins or growth factors. And these signals tell the cells, again, whether they're being delivered or whether they're migrate to the wound injury. They tell the cells what to do and how to repair the tissue. And then you have the scaffold that combines everything together. And here you can vary this scaffold to have different geometry, have different porosity, and also to degrade at different time scales or through different mechanisms depending on your application. And when you combine all three of these, this is called the tissue engineered construct. You don't necessarily need to have all three of these types of components, but some combination. And you select them, each of these depending on your specific application. And so in terms of what my lab does is we really focus on the scaffold part. So how can we design materials that really closely mimic the extracellular environment, so the local environment that the cells see, and directly interact with cells to help them regenerate functional tissue. We've done a lot of work in what I'm going to call kind of homogeneous or more single tissue repair. So things like you're just repairing one tissue, so bone or meniscus. More recently, we've been moving towards replicating more complex signals. So how can we kind of control in both space and time how the cells are behaving? And so this requires you to have more dynamic control over your material properties. And so how do we do that? I'm going to first talk about how we've done this with signal ingredients, so how we've been able to control structure and chemistry in space. We're really interested in spatial control over biomaterials because in particular, we do a lot of work in orthopedic tissue engineering. And when you consider these types of tissues, the musculoskeletal system is really connected and complex. And so you can see this picture of this man walking or running. And as you move, you're transferring load from your muscles to your ligaments and tendons to your bone and back again. And as the tissue transitions from one tissue type to another, you have what's called interfacial tissue in between the two different tissue types. And at these interfaces, you're changing in chemistry, biology, structure. And so these transitions are really, really important for everyday motion and movement and functionality. Unfortunately, these tissues, these transitionary tissues, are also commonly injured, right? And so it's really important to be able to spatially control cell behavior to kind of mimic these transitions so that we can better clinically repair them. To kind of give you some examples of what these interfacial tissues look like, here are a couple of examples of cartilage bone, muscle, tendon, and meniscus bone, all of which seamlessly transition from one tissue type to another tissue type. And as you can see here, we can even take the cartilage bone as an example here. It's going from a superficial cartilage, which has one type of fibrous structure, to the deep zone, which has a different type now of fibrous structure. And then it becomes calcified cartilage. So there's some additional mineralization in that cartilage before it transitions to the bone. And these transitions are really, really important for load transfer and for functionality. We're going to, the specific interface that we're going to focus on for this first part of the talk is the tendon-bone interface. So this interface is really common. The Achilles tendon to bone interface can be commonly injured, but the even more common injury is the rotator cuff. So rotator cuff injuries are really, really common. That's an injury in your shoulder. These injuries typically occur at this tendon-bone interface. And so you can see here that tendon tissue is really highly aligned. It typically has fibroblast cells, and it's more collagen type 1. And as it transitions to bone, you start to see this mineralization, highly mineralized tissue, and it has more osteoblasts. And this transition from this highly fibrous tissue to this highly mineralized tissue occurs over around 500 microns. So you're able to kind of gradually transition from this one tissue type to another tissue type over these relatively short distances. A lot of different work has gone into trying to recreate these types of structures and gradients using a bunch of different manufacturing techniques. Here are just a few examples of them. In particular, a lot of work has focused on the use of electrospinning for this. However, it's been difficult to separate and be able to independently control both the chemistry and the structure while maintaining this really high resolution of less than a millimeter in terms of going from one state to the other state, so aligned to unaligned. One of the reasons that electrospinning has been wildly used is because it does allow you to create materials that have this fibromorphology that mimics that of native tissues. So if you're unfamiliar with electrospinning, the way it works is you have a polymer solution and you apply a high voltage to the polymer solution. As the charge from that voltage in the solution exceeds the surface tension at the tip of the syringe, you'll get this Taylor cone to form, and then the polymer will be ejected from the syringe. The solvent will evaporate, and then the dry fibers will collect onto a grounded collector. You can have a bunch of different types of grounded collector. Here I have a rotating mandrel shown, but you could also have a flat plate collector as well. And as you can see, a lot of our tissues in the body have this fibrous morphology. So cardiac tissue, tendon tissue are shown here, as cardiac tissue having a less aligned structure and tendon tissue being really highly aligned. And what's nice about the electrospinning is that it does recreate this fibrous morphology really easily. And so here is showing that at low rotation speeds on this cylindrical collector, you have these unaligned fibers, and at really high rotation speeds, you're able to collect highly aligned fibers. And so by changing the mandrel rotation, people have been able to tune the alignment within their scaffolds. The other reason we really like the concept of using electrospin fibers for these types of applications is, as you can see here in this schematic of the shoulder joint, the supraspinatus tendon, so that's the rotator cuff tendon, here is attached to the shoulder bone, the humerus. When you have a rotator cuff tear, the surgical repair is to suture the tendon back to the bone. And what's nice about this is that you can use the electrospun fibers as a patch during this repair procedure, and that this has actually been done clinically in humans before. This is a collagen patch and a polypropylene patch that have been implanted into human rotator cuff joints. Unfortunately, these patches have not had a lot of success, and I would argue that the reason they haven't had a lot of success is they don't actually mimic that tendon to bone tissue. And so here in this case, they're really just being added for mechanical augmentation, but that if you want to have improved regeneration, that you really need to mimic that tendon to bone interface so that you can serve as a template for the cells that are there to regenerate new tissue. But the benefit here is that these procedures, the clinical implantation procedures already exist. And so we don't need to create new procedures using these electrospun fibers patches. And so as I mentioned before, you can very easily control fiber alignment using mandrel rotation speed. This has been really well established. So if you're collecting onto a flat plate, or if you're rotating, very, very slowly, you'll have randomly aligned fibers. And as you increase your rotation speed, they'll be very nicely aligned, and you can really tune this with your mandrel rotation speed. People have controlled the alignment during electrospinning. So you can create gradients very easily by changing the electrospin parameters with time. And so, for example, you can start out with one rotation speed. And so your initial layers that will be deposited will be randomly aligned. And because electrospinning is a layer by layer deposition process, so if you start out at this slow speed, your first few layers will be randomly aligned. And then as it's depositing more layers with time, you can change your rotation speed so you can increase it. And so your fibrils will start to be semi-aligned in the middle. And then if you further over time increase your rotation speed, you can start to get these highly aligned fibers. And this or other similar techniques have been done to either change fiber alignment. You can change polymer concentration this way by changing the concentration of the solution with time, and a whole host of other process and parameters can be changed with time. The problem with these types of approaches that depend on creating gradients by changing the parameters with time is that you're creating here gradients in the depth of the scaffold. So you're not actually creating gradients within a single layer. And so there's really a need to develop new electrospinning methods that allow you to control the fiber alignment along the length that kind of mimics that tendon to bone interface, and also allows you to control the alignment and the chemistry within a single layer. And so we turn to magnetically assisted electrospinning to do this. This has been done in the past to create highly aligned fibers. And you can see here that it is capable of producing these really nicely aligned fibers in the presence of a magnetic field. And they're just using permanent magnets here to create this effect. So we hypothesized that we could then really control where the magnetic fields were. And so we could then use this as a technique to control where we were able to get fiber alignment and where there was not fiber alignment, in order to, again, create these fiber alignment gradients that mimic that tendon to bone interface, and then can serve as a template for the cells to regenerate tissue with a similar organization. So here's our setup. So we have a flat plate collector. In this case, the magnet is shown in orange and denoted with M, and then the non-magnetic region is shown in green and denoted as NM. And you can see that we are able to get really highly aligned fibers in the magnetic region, and then it transitions to non-magnetic, or to randomly aligned in the non-magnetic region. We also have this really nice transition region in between the two. We can quantify the fiber alignment in each of these areas using image J. And here you can also see the distribution of those fibers in each region. You can see the smooth transition from the highly aligned region to the randomly aligned region. What's really exciting about this is that the scale bar in that macroscopic image in the upper corner is 1 centimeter. So we're transitioning from highly aligned to randomly aligned in less than 1 millimeter, which again, if you recall, I noted that the transition from highly aligned to bone occurs in around 500 microns. So this transition is really important that we're on that length scale. So we wanted to see how versatile our method was, and so we tested it with a range of polymer materials. And so we were able to get high fiber alignment in the magnetic region using polycaprolactone, polyethylene oxide, polyethylene glycol, very common biomaterials used for electrospinning, as well as cellulose, which is a more biological biomaterial. We were not able to get fiber alignment in the magnetic region for alginate and HA, hyaluronic acid. So in terms of what we believe is driving fiber alignment here, what I want to note is that we did not need to add magnetic nanoparticles to any of these solutions. So the alignment is occurring due to the inherent magnetic susceptibility in each of these materials. We believe that the charge on alginate and HA is disrupting their ability to align in the magnetic field, and is disrupting that electromagnetic field close to the magnet. Because if you recall, right, during electrospinning, there's a high voltage that's applied. And so there's the combination of both the magnetic and the electrical field is really, really important that in order for alignment to occur. And so we believe with these charged polymers that unfortunately that does inhibit their ability to align in that electromagnetic field. And I'm happy to talk about that, about what we think is going on here more. But what's nice is that we do see it for a wide range of polymers, both synthetic and for the cellulose, for a biological material as well, that we are able to get high fiber alignment in that magnetic region. So next we wanted to see how long we could maintain fiber alignment. And so we're using really low magnetic field strength, so on the order of millitesla. And so that magnetic field strength does dissipate quite quickly as you get away from the magnets. This is why we see the transition from aligned to randomly aligned occur so quickly, is because of how quickly that magnetic field dissipates. What this also means is that as the layers are deposited on the magnet, the new layer is further and further away from the magnet. And so as you build up that scaffold over time, you're also dissipating the magnetic field strength at that new layer thickness. And so we also see this in terms of how long we can spin and maintain fiber alignment. We're able to maintain fiber alignment up to about 4 or so hours. This again corresponds to around that 500 micron distance away from the magnet. And once we get to about 6 hours, we start to see that the fibers are now not aligning anymore over the magnet. That being said, of course, you could stack if you wanted a thicker scaffold, you could stack multiple aligned scaffolds on top of each other. And so there are ways to create more 3D structures if needed. We wanted to see if we could create more complex fiber alignment gradients. And so here we first put 2 magnets close to each other. And you can see here that by putting these 2 magnets close to each other, labeled M1 and M2, we're able to extend the region that is highly aligned. And again, it transitions to this randomly aligned region in the non-magnetic region away from the magnet. We can also place the magnets far away from each other. And in this case, as you would expect, they align over each magnet. But if those magnets are far enough away from each other, they will be randomly aligned in between the magnet. And so this just demonstrates how much we're able to really control within a single layer where in space we're getting fiber alignment. Next, we were interested in if we could control fiber alignment direction. And so here we're looking at an L and a T shape. What's interesting here is that if you look at this T shape, for example, you'll notice that the fibers are oriented horizontally in the bottom part of the T, and then they turn 90 degrees and are oriented vertically in the top portion of the T. So we're able to control not only where in a single layer we're getting fiber alignment, we're also able to control the direction of that fiber alignment. Next, we were interested if we could control the shape of the interface between the magnetic and non-magnetic regions. And we were able to do that quite easily by using foil, again, to disrupt that electromagnetic field. And we can very easily machine or simply cut the foil into the desired shape. And so here we're showing it with 1 centimeter amplitude waves. And we get this wavy interface between the aligned and randomly aligned regions. We are really interested in particular in this type of wavy interface because the natural fiber cartilage or tendon to bone interface is wavy and interdigitated. And this interdigitated interface is really important for load transfer between the tendon and the bone in order to minimize stress and strain concentrations at this interface and in order to reduce the risk of failure. So next we seeded some mesenchymal stromal cells onto these materials, and we looked at their morphology and their gene expression. And as expected, the cells are aligned kind of in the direction of these fibers in the highly aligned region, and they're more, they have less directionality, and they are more spread in multiple directions in this non-magnetic region. And we can characterize this using aspect ratio and then by also looking at what direction they're oriented. We also looked at tendon-associated genes. Here's just an example of one of those genes. Scleraxin has kind of increased gene expression at 7 days on these aligned fibers. And all of this was expected. Other groups have noted that fibers on, when seated on aligned fibers, will spread like tendon cells in this aligned manner, and that fiber alignment is sufficient to induce tenogenesis. So next we wanted to look into how to create chemical gradients as well. We looked into two techniques, offset electrospinning and a technique called post-electrospinning photoconjugation. I'm not going to go too much into offset electrospinning, but I'll just say that we found that technique had limited spatial resolution, that it was only able to control the chemistry gradient on the order of a centimeter. And as we mentioned, we really want sub-millimeter resolution. And so I'll just go ahead and continue to talk about what we've done for the post-electrospinning photoconjugation method. So the way this works is you need a photoreactive polymer. In this case, we're using Norbornean here. And so Norbornean we really like for this application because it allows you to really controllably add molecules or cross-linking to your scaffold, since the Norbornean groups themselves will not react on their own. So you can make sure that you have additional Norbornean groups available for further cross-linking or conjugation to other molecules. And so the way this works is, so you have a Norbornean functionalized polymer and you swell in a biomolecule. So in this case, you would swelling a biomolecule with a thiol group on it. So if it's a peptide, you could use a cysteine residue that would have that thiol group on here. And then in the presence of UV light, those thiols will react with the Norbornean groups. And so you can control which parts of the scaffold are exposed to UV light and for how long. And then you can wash away the unconjugated biomolecules. Since the tendon bone interface goes from the biggest chemistry gradient, the one that's of kind of most biological significance, is the mineralization chemistry gradient, we're interested in how we can do that with these scaffolds. So one way to do that is to use a calcium binding peptide. So if we're able to create a chemistry gradient of this calcium binding peptide shown with the sequence shown here, then we can take this scaffold and immerse it in a simulated body fluid in order to promote in vitro mineralization, and then wash away whatever the unbound minerals are. And then ideally here we have this scaffold that has the highly aligned to mineralized bone gradients that mimic that tendon to bone gradient. So we were able to create these gradients using an in-house designed sliding photomask that allows us to create a lot of different types of gradients, from single gradients to multi-directional gradients. We're also able to really nicely control our mask speed, and by controlling the mask speed, we're able to have very sharp gradients of around 250 microns to larger gradients of over a millimeter. And so when we add this peptide and then perform the in vitro biomineralization, we can then visualize the amount of mineralization on our scaffolds using an alizarin red S stain. And so you'll see it here shown in red. And then we can quantify how much of that stain is on our scaffold to get an idea of the mineralization as a function of with or without peptide, and for how long it was in the mineralization solution. And so as you can see here, that without the peptide, we have minimal mineralization, and with the peptide, up to an hour, we're able to get good amounts of mineralization without having mineralization on our control scaffold. And we can then further apply this using our in-house designed sliding photomask to then also create a gradient in mineralization as well. And so we can combine this with our control with fiber alignment. And so what we're doing now is using these dual alignment scaffolds that have alignment both in fiber alignment and mineralization, and evaluating them both in vitro and in vivo more deeply in order to understand how not only the cells behave in response to this gradient, but if they can be used to better heal rotator cuff injuries. We're also looking at a lot of kind of other different projects kind of in this spatially controlled area. So looking at other different types of peptide gradients beyond the calcium binding peptide, looking at ways to create 3D structures, similar to, as I mentioned before, either through stacking multiple scaffolds together, or here I have an SCM of a rolled scaffold, as well as other applications. So the next part of the talk, I want to spend talking about ways that we've tried to control biomolecule signaling in time. And so hopefully I've already been able to convince you why controlling biomaterial properties in space is important, but it's also really important to control them with time. One of the reasons this is important is because when you have an injury, or when there's you have a disease, this signaling cascade is really complex. And so you can see here, this is the signaling cascade during bone repair. And this is just a few of the molecules here that are expressed during bone repair. But in general, you can see that there's this inflammation period followed by immature regeneration before you start to have more mature regeneration. And in response to these different periods of healing, you have signals that turn on and then turn off and then turn on again, or vice versa. But you have this really complex, very dynamic, very cyclical time course in terms of when these molecules are on and off. A lot of different groups and chemistries have been utilized to try to mimic this complexity, from covalent bonds to non-covalent bonds to the use of external stimuli to turn molecules on and off. And in particular, there's been some really elegant work, especially out of the DeForest lab at the University of Washington, that's been able to really nicely show that they can turn molecules on and off multiple times and with multiple molecules. One of the downsides to a lot of these approaches is that they require these really complex conjugation schemes, and typically you need a different conjugation scheme for every molecule that you're trying to turn on and off. So we wanted to develop a new platform that was easier to use. And so we, when we were thinking about what would be in the ideal platform, these are the design considerations that we came up with. You need to have spatial control in order to mimic the spatial heterogeneity of many tissues. You need to be fully reversible over multiple cycles to mimic the temporal complexity of these signaling cascades. You want to have orthogonal control over multiple molecules so that you can evaluate the synergy or the role of different molecules in healing. And you want it to require a single conjugation scheme and be easy to use so that other labs can easily use it and apply it to their applications. And so we thought to use DNA as a reversible linker for this platform as a result. And so we're going to use here DNA as the handle to turn molecules on and off. And I'll get into more about how we're going to do that in a second. And then the base material, we decided to use hyaluronic acid as the hydrogel material. And we chose hyaluronic acid as it's a natural polysaccharide. It's present throughout the body and interacts with a number of cell receptors. It also has synthetic versatility, so it can be easily modified via these carboxylic acid and hydroxyl functional groups. So we can add that Norbornian group that we mentioned before, and again, utilize this Norbornian thiol chemistry to tether molecules onto the surface. And again, we've already demonstrated that we can use this Norbornian thiol, and again, kind of shown here, to really spatially control the tethering of different molecules onto these hydrogel surfaces. So then we're going to use DNA to turn molecules on and off. One of the reasons we really like using DNA as a reversible linkage in this case is because it has really high specificity. So you can have multiple different DNA strands, and you can have different DNA strands for each biomolecule that you want to tether on. And each set of complementary DNA strands won't bind to the other set of complementary DNA strands. Okay. And so the way that this platform essentially works is we have our hydrogel here that has the Norbornian functional groups. We tether single-stranded DNA to the hydrogel surface using that thiol Norbornian chemistry. And we can tether this initial surface strand to the hydrogel via photo patterning. And so that enables us to spatially control the location of this initial surface strand, okay? So this surface strand will have a specific sequence. And so we can add the biomolecule that is tethered to the complementary strand to that surface strand. And so through DNA hybridization, those two molecules will bind to each other. And so we'll have our biomolecule on now the hydrogel. So the signal will be on. You'll notice here that this biomolecule strand has this orange DNA sequence that is not bound to the surface strand. So what we do is this biomolecule strand is 5 nucleotides longer than the surface strand, and this is called the toehold region. And so when we add a displacement strand that contains those 5 additional nucleotides, those nucleotides outcompete the surface strand because it matches the biomolecule strand more closely. And so it will now bind to the biomolecule, and then you can wash away the biomolecule bound to the displacement strand. And so now you're just left again with this surface strand that's still attached again via that thiol Norbornian chemistry onto the hydrogel surface. And so you can repeat this cycle over and over again as many times as you would like. So here's just how we're adding we can really easily tether bioactive peptides to the DNA using copper-free click chemistry. And then again, in terms of just the nomenclature that we're using here, the surface strand that is covalently tethered to the hydrogel, we call S. The complementary biomolecule strand with that toehold region is called CB for complementary biomolecule. When we add that, the signal is now on. And then we have a displacement strand that is complementary to the entire sequence plus the toehold region, and that's called complementary displacement. And when that is added, it removes the biomolecule. Now the signal is off. We also have mismatched biomolecule and displacement strands. So these are strands that are not complementary, and so they're marked with MB and MD. So to walk you through how this, to kind of demonstrate to you that this system works, we first have the surface strands on, we add our complementary biomolecule, so the signal is on, and so you can see this really high intensity because the signal is on. You see this nice red image because we use a fluorophore to demonstrate that it's tethered. We then add the complementary displacement strands, and you can see that now that fluorescence intensity goes all the way down, and so the signal is off. If instead we have our complementary biomolecule added to the system, so the signal is on, but instead of adding a complementary displacement, we add a mismatched displacement, you'll see that the signal stays on because the displacement strand is not complementary to the biomolecule strand. And if we have our surface strand and try to add a mismatched biomolecule strand, you'll see that the signal does in fact stay off, so that the biomolecule strand needs to be complementary to that surface strand. And if we try to add a biomolecule strand without a surface strand, we also don't see any signal, demonstrating that there's no non-specific binding occurring onto these hydrogels. We can turn these signals on and off over many, many cycles. Here's shown 6 full cycles. And when we add these DNA and biomolecules to the surface, we're not impacting the modulus of these materials, which is really important because modulus has been shown to impact cell behavior. We can also have independent control over multiple biomolecules using the same DNA conjugation scheme. So if we have tethered 2 different surface strands onto our hydrogel that are complementary to 2 different biomolecule strands, that allows us to have a red and a green signal. And when they're both tethered onto the surface, we see this yellow signal due to both of their presences. We can add the displacement strand for red, so removing the red signal. And so we're left with just the green signal. And then we can add the displacement strand for the green signal, and in the same solution, add back in the red signal. And so you see here that we're able to switch out the green and add in the red very easily. We can also do this with spatiotemporal control, again, because those initial surface strands are tethered to the surface using UV. And so we're able to have the red and the green signal with spatiotemporal in this crosshatch pattern shown here. We can remove the red with the red displacement strand, and then remove the green and add with the green displacement strand and add back in the red using the red biomolecule strand. And we can, in fact, do the exact same thing using our sliding photomask. And this is, again, the same scheme that I kind of presented previously. We have the overlapping multi-directional gradients of red and green. So we have high green here, high red and green, and then no biomolecules in each of these quadrants. And you see the intensity with distance to kind of show that they are in fact a gradient. And we're able to, again, remove the red and then remove the green and add back in the red very easily. So in terms of how much molecule we're able to get onto these surfaces using this technique, we've been able to, here we can show that the amount of RGDS that we're tethering is equal to the amount that is in the solution. And also it's comparable to without the DNA. So here we have RGD, which is a cell matrix adhesion peptide. And so when we add the RGD without the DNA, so when we tether it directly to the hydrogel surface, compared to when it's tethered using the DNA, we see similar cell areas, so spreading of those cells with and without the DNA. And of course, as you would expect, as you increase the RGD concentration from 0 to 1 millimolar, you see increased cell spreading, as you would expect for the inclusion of RGD. But we're really happy to see that the DNA did not inhibit or enhance this cell spreading. We are also interested in looking at osteogenic growth peptides. So this is a naturally occurring peptide and is known to regulate proliferation, differentiation, and mineralization. And so you can see here that when we add OGP onto our hydrogels, here's just cells on our hydrogels in growth media, cells on the hydrogel and osteomedia, and then cells on the hydrogel and osteomedia with 1.1 nanomolar OGP. And this staining here is blue is for alkaline phosphatase expression, you can see this increased alkaline phosphatase expression in the presence of OGP. We also can look at this as a function of OGP concentration. And so we use this to select the best OGP concentration moving forward. Here you can see that these low OGP concentrations, 0.1 and 1, do have a slightly higher ALP expression compared to the highest OGP concentration that we looked at of 10. And the reason for this is that these higher OGP concentrations actually start to have a deleterious effect on the cell behavior. And so we selected these two lower concentrations for the next study where we wanted to look at the temporal effects of OGP presentation. And so what we did is we cultured cells for 14 days on our materials. And in one experiment, we had OGP on for the first 7 days, turned OGP off, and then continued to culture until day 14. In the second set, we had OGP off for the first 7 days, and then turned OGP on at 7 days until day 14. And in a third experiment, we had OGP on for the entire 0 to 14 days. And so we're comparing those three groups here. The early presentation is shown in black. In the middle is when it's presented the entire time, so constantly. And on the right is when it is presented late or delayed. And what's interesting is that the delayed presentation of OGP has the highest ALP expression, showing that the timing at which you're turning on these osteogenic signals is really important. So hopefully I've been able to show you that we've been able to develop this new DNA-based biomaterial platform that allows us to have independent control over multiple biomolecule signals in both space and time. I've been able to demonstrate and control these biomolecules spatially using the Norbornene thiol chemistry, demonstrate full reversibility over multiple cycles, orthogonal control over multiple biomolecules using this single DNA conjugation scheme. And because of this, it makes this platform really easy to use and translate to other applications. Future work is combining this DNA approach using the electrospinning techniques that I presented in the first part of this project. And here is just a quick look at that. Here's some images of the fluorescent DNA on these electrospun fibers. And so with that, I'm going to conclude my talk, and hopefully I've been able to show you a lot of the different techniques that my lab is using to both spatially and temporally control biomaterial properties for tissue regeneration. And with that, I would like to acknowledge my lab members, particularly Fallon and Kevin, that did a lot of work that I presented today, as well as my funding sources and collaborators, especially Nicholas Stephanopoulos, who has really contributed a lot of his DNA expertise for that second project that I presented on. And with that, I will take any questions.
Thank you very much for your presentation, Dr. Julianne. If anyone wants to ask something, you can use the chat window within the Cisco WebEx application. So let me try to understand this kind of challenge. You present at the beginning of the presentation an image about the working body. You know, it's moving, have a different challenges. Do you have in mind to evaluate the biomechanical response of the materials? Because you have the gradient strategy to replicate the way that these systems have in the body, but they have to respond to a mechanical specific value. I don't know if I'm trying trying to be clear, but I'm not achieving that. But the question is simple. Do you have biomechanical experiments in that kind of material, the first part of the seminar?
Yeah, that's a great question. So we have evaluated the mechanical properties of the aligned and randomly aligned regions. And maybe to take a step back, certainly cells do respond to material stiffness, and that can influence cell behavior. It's a little bit complicated in the sense that cells are really sensing local mechanics, right? And not necessarily bulk mechanics.
Yeah.
And so it's not as easy as measuring the bulk mechanics of your system and saying that that's what the cells are feeling. That being said, right, bone is much stiffer than tendons are, right? And so you would want to design a system where your bone region is stiffer than the tendon region. And that was one of the reasons why we were really interested in creating a mineralized compartment for the bone region. In the sense that we hope that that mineralized compartment adds additional stiffness, because of course aligned fibers are much stiffer than unaligned fibers. So if you take just the aligned to the randomly aligned scaffold, the stiffer part in that gradient scaffold is actually the tendon compartment and not the bone compartment, right? And so yeah, so that's why we were most interested in adding that mineralization compartment or mineralization component to help with the stiffness there. All of this being said, the idea is that it serves as a patch, and that we're providing more chemical and physical cues as in structural cues for organization, and not necessarily as to have mechanical augmentation. Okay. And so the idea is not that it will carry load, because we don't think that it's not going to be strong enough to carry load, right? That's not the purpose. Okay. But that it can be as a patch during the healing period, and that you regenerate new tissue that is able to carry the load, if that answers your question.
Yeah. In that sense, do you calculate the time that these scaffolds will be used by the body to regenerate the tissue and the bone and everything? Do you have any calculus about that, estimation of the time that the parts the prostheses are going to be working in the body?
So I can definitely give you an answer in terms of so we're looking at small animal models right now, and that time scale is on the scale of 4 to 12 weeks. And I think that the human time scale would definitely be a little bit longer than that, probably 2 to 4 months instead. That last part is a guess, but because we've been more focused on what the small animal model time scale is and working with that right now. But yeah, so I think the idea would be that you would want a material that would stick around probably for 8 to 10 weeks, most likely. Okay.
And so the design of this tissue is aligned, this not aligned, is answering a specific situation in the body, or you are just trying different paths in other, the L shape, the T shape is to study the response of the material maybe, but that L shape, that T shape is thinking about that situation and a specific situation.
So we're primarily interested in this tendon and bone application, which is just goes straight from aligned to randomly aligned. It has that interdigitated kind of wavy interface in between. The L and the T was more just to demonstrate the versatility of the method for other potential applications that people may be interested in. But we haven't thought too much about how we could use kind of those other kind of more complex arrangements yet.
Okay. So Dr. Victor Alcaraz is asking in the chat window, thank you very much for your brilliant seminar. Meniscus injuries are very common in athletes. Is this technology already applied in some sports?
Yeah, unfortunately, there are no tissue engineering therapies for any orthopedic injuries, really. They have not had much success. It's really hard to regenerate or repair tissue for a whole host of reasons. One, the mechanical loading makes it quite difficult. So these tissues are typically really dense. There aren't a lot of cells there. And the tissue is really dense. And so the cells that are there, it's hard for them to kind of move around. So I think what we've been trying to do is to kind of create a pro-regenerative environment that is less dense, that cells are there for, and then provide those physical cues so that they can regenerate the tissue that's needed and that tissue is functional. So I think, yeah, hopefully we can see some of these therapeutics in the clinic at some point, but I think it's still very much a developing technology.
Okay. Omar Rogelio Gomez is asking, well, hello, Dr. Holloway. Thank you for your talk. The molecules engineered in your work can be engineered to be more effectively mimic natural proteins, such as the hierarchy. I don't know how to read that. Maybe you can.
Yeah, I have it up. The
yeah, you can,
hierarchical clustering, I think.
Yeah, of PA and nanofibers with the DNA to mimic the assembly of collagen. What is the biggest challenge to achieve the goal? I don't know. Is the question clear enough for you?
Yeah, so we do work with Nick Stephanopoulos a lot. He's done some work in this area. He did his postdoc at a Sam Stoops lab, and they've done a lot of work in trying to do these kind of biomimetic self-assembled fibers to kind of mimic the structure of collagen. So I certainly think there's a lot of work there that is being done in terms of how to create these how to kind of mimic the assembly of collagen. And I think that's certainly and I think Nick is still definitely very much working in that field. We're more just interested in and have been focused on how to control biomolecule presentation and control. But I think certainly that's a good point, that I think some of these techniques could also be combined, right? And that would be pretty interesting as well.
So maybe that's the last question for you. I would like to thank you the opportunity to that you had the chance to share your work, your impressive work. Really is amazing the way you are approaching this kind of challenges in the medical area. And also this is a very good example of how the chemical engineering can be applied to a specific field. But in order to achieve that kind of goals, you have to work with a very large group of people and specialists, and you're a role model to us. Really, we are very glad to have you here.
Thank you. I appreciate that very much, and it's been a pleasure to present with all of you today.
Thank you so much. So I'm going to stop the recording.